to Biomix Hub. In this today's video, we will discuss about the dorsal ventral axis formation in case of Drosophila. In the last video, we have already discussed about the basics as well as anterior posterior axis formation in Drosophila. Now let's get started with the video. So in case of Drosophila, the dorsal ventral axis formation takes place before fertilization and in the oocyte and after fertilization. Okay, after fertilization. So what happens in the egg, there are nerve shell and oocyte which are present. This is the egg. This nerve shell, what is this? It provides nourishment to the cell, and in the oocyte there is a nucleus and follicular cells are surrounded. Okay, these follicular cells, they are not present in the nerve cell. So during the development, certain development, there are cytoskeletal rearrangement. When the oocyte is enlarging, there are cytoskeletal rearrangement, due to which the nucleus which are present in the oocyte, it is being transferred to anterior dorsal side. Okay, this is the anterior dorsal side. So this is the first case where it is deciding before fertilization that this portion is the dorsal side of the upcoming Drosophila. Okay, so this is the enlarged portion of this part. So here we can see these are the follicles, these are the follicle shells, and here the nucleus is being transferred to anterior dorsal portion, and near it the Gherkin mRNA are present. Okay, the Gherkin mRNA are present near the dorsal portion, and here after the formation, when the nucleus enters, when the nucleus comes near it, after the fertilization, it is being converted into Gherkin protein. If it is a protein, it can be a transcription factor. This protein needs a receptor. So the receptor for this Gherkin is present in the follicle shell, which is known as torpedo. Okay, and this torpedo which is present, it is present both in the dorsal side as well as in the ventral side. This torpedo is being present. Okay, these are the receptors which are present both in the dorsal as well as in the ventral side. Because this Gherkin protein cannot be synthesized into the long distance, that is the reason it cannot travel near the ventral side. Okay, so here as it is nearer to the dorsal side, it will come and bind, the Gherkin protein will come and bind near the dorsal side, come and bind with the receptor that is known as torpedo, and it is in the dorsal side. Okay, so when this torpedo is binding, the Gherkin is binding with the torpedo, there needs some changes. There is a protein which is known as spike protein which is present in the follicle shell. So when there is interaction, there is inhibition of pipe protein in the dorsal side. So here there is inhibition of pipe protein in the dorsal side, but in the ventral side there is no pipe protein inhibited. There is synthesis of pipe protein in the ventral portion. Okay, so what next? Here what happens, here the dorsal fate is being determined after this Gherkin protein has binded to the torpedo receptor. It has decided the dorsal fate and it is inhibiting the pipe protein, and this pipe protein is responsible for ventralization of the Drosophila. Okay, so here the pipe protein is being synthesized. Now we will see here in the ventral region. So the pipe protein is being synthesized in this region. So this pipe protein, after it synthesized, it activates some other cell. Like we can see that this is the embryo and it is having a vitelline membrane. Here vitelline membrane is present, and this is the pre-vitelline space, and here the cells which are dorsalized cells, these are the dorsalized cells which are present, and these are the ventralized cells which are present. Okay, these are the ventralized cell and these are the dorsalized cell of the embryo, and this is taking place in the fertilized egg. Okay, so when the pipe protein is synthesized, it is activating some ventralized vitelline membrane proteins, which are after the phosphorylation, the GD is being attached with it. Okay, what this GD does, this GD which is present, like we can see here, the pipe protein sulfates ventral vitelline proteins. There are ventral vitelline proteins which are present, which the pipe protein will be, it will help in the sulfation. Here the sulfation is taking place. Next, sulfated vitelline membrane proteins bind gastrulation defective, GD. These are the sulfated signal proteins which is being sulfated by the pipe protein, and this is being bounded by the GD protein. Okay, what is the work of this GD protein? This work of this GD protein is that it will cleave the snake protein. Okay, It's cleaved snake protein, because the snake protein was not activated. When it will be cleaved, then it will be activated. Okay, now it forms a complex. Another one protein comes which is known as the ester protein, which is in the uncleaved form. So now this forms a complex. Okay, this is the pi protein which is sulfated, this is the GD protein, this is the cleaved snake, and this is the uncleaved ester which forms a complex. And after this, the ester protein is also cleaved and it is activated. Now this ester protein, when it is activated, it comes and binds with the spazole, okay, which is now in the uncleaved form, and ester will help in the cleavage of the spazole protein. And when the spazole protein is cleaved, it will be activated. Then it will bound with a receptor which is present, which is known as toll receptor, which is present in the vitelline, which is present in the pre-vitelline membrane. Okay, so here what this does, it will help in activating this tube and pelle. What it will do, it will bind with this tube and pelle. After the toll receptor is being activated, it will bind with tube and pelle. So what was the work of this tube and pelle? So this was one case. Now the next case is in the ventral side, there was mRNA which was known as dorsal mRNA, and after fertilization it was forming the protein, dorsal protein. And this dorsal protein, this is a protein which will help in the ventralization of the embryo. Okay, this dorsal protein, when it enters inside the, and when this dorsal enters inside the nucleus, it will help in ventralization of the embryo. But the dorsal is being regulated by another one protein which is known as cactus. This cactus, it will bind with the dorsal and it will not allow the dorsal to enter inside the nucleus. Okay, so there cannot be ventralization, proper ventralization of this embryo. So this pelle and tube which are present, they will be activated and they will bind with this cactus protein. Okay, and they will phosphorylate it. When they will be when it will dorsal and cactus protein, and they will phosphorylate it. Okay, so what happens when the cactus is being phosphorylated? It cannot bind with the dorsal protein, due to which now dorsal protein is free and it can enter inside the nucleus. So this nucleus which are present, these are the nucleus which are present in the vitelline membrane. So this dorsal protein will enter inside this nucleus and it will help in ventralization of the embryo. This is how before fertilization and after fertilization the dorsal ventral axis formation is taking place in case of Drosophila. I hope you have understood this video. Oh, thank you everyone for watching this video till the end, and if you like this